Welcome to my channel, and this video is about the DNA binding domains, and we are going to discuss their types. So a DNA binding domain is an independently folded protein domain that contains at least one structural motif that recognizes double or single stranded DNA. DNA binding domain doesn't disrupt the base pair of DNA, but it changes the configuration of the backbone by binding the DNA double helix. So can be defined as it is a part of the DNA binding protein which is specialized to recognize and bind to a region of the DNA sequence. So motif are basically pattern of super secondary structure in proteins, combination of the alpha helix and the beta sheets. So it can be categorized into four types. First one is the helix turn helix motif, helix loop helix motif, leucine zipper motif, and the fourth one is the zinc finger motif. So HTH was first DNA binding structure to be identified in the bacteriophage repressor. So two alpha helices are separated by a beta turn made of a four amino acid. So it is also called as a recognition helix, and figure are as follows. It's a term that is homeodomain. It is an extended HTH motif found in protein coded by a homeotic genes. So second one is the helix loop helix motif. It is a conserved domain involved in a DNA binding and a protein dimerization found in the eukaryotic regulatory protein. It consists of the 40 to 50 amino acid comprising of two amphipathic alpha helix and a loop of variable length linker. So figure are as follows. It can be like first figure, or it can be like loop like structure which forms the N terminal and the C terminal separately. So third one is the leucine zipper motif. It has a two separate domain which mediates both DNA binding and the dimerization. So figure shows the leucine zipper motif. As figure describes the hydrophobic side, the hydrophilic side, leucine residues, and the DNA major groove and the minor groove. So zinc finger motif, a group of the DNA binding domain includes one or more zinc ion as a structural components. So finger motif was first discovered in a transcription factor of the Xenopus levis in the transcription factor 3A, and it is essential for the correct folding of the DNA binding. So figure are as follows, like for two cysteine residues and the two histidine residues connected by the zinc. So these are the description of the DNA binding domains and their types. So keep watching. Thank you.